Darwin pointed out that both frigate birds and upland geese have webbed feet, yet neither goes into water. He explained their feet is a leftover from their past, both being descended from waterbirds. Evidences from comparative embryology. One of the most fascinating sources of evidence for the validity of Darwin's theory of evolution has come from studies of comparative embryology. The suspicions of a common ancestry for very diverse sets of organisms from comparative anatomical studies have been supported by embryological data. As Darwinian evolution suggested that more complex organisms would have achieved their state by elaborating on the existing developmental patterns of more primitive forms, so one would expect to find that certain relatively simple organisms and more complex ones have many initial developmental steps in common. In fact, the more developmental steps two species have in common, the more closely related they are to a common ancestral form. Comparative embryological studies have revealed that there was one developmental pattern that could be viewed as having undergone a series of branchings. All multicellular animals start their development as a single zygote, and through a series of meiotic divisions, increase in cell number until a blastula is formed. The developing embryo elaborates upon the blastula stage by forming two fundamental germ layers, ectoderm and endoderm, during the course of gastrulation. After the differentiation of the ectoderm and endoderm in the gastrula, the third germ layer, mesoderm, is formed. There are three distinct patterns by which developing embryo of different species produce a mesodermal layer. For instance, in annelids, mollusks and certain other invertebrates, the mesoderm develops from special cells which are differentiated early in cleavage. These cells migrate to the interior and come to lie between the ectoderm and endoderm. They then multiply to form two longitudinal cords of cells which develop into sheets of mesoderm between the ectoderm and endoderm. In primitive chordates, i.e., amphioxus, etc., the mesoderm arises as a series of bilateral pouches from the endoderm. These lose their connections with gut and fuse one with another to form a connected layer. The mesoderm in amphibia, reptilia, birds and mammals is formed from the cells which remain associated with either ectoderm or endoderm or both and which get disassociated from them to migrate and come to lie in between both germinal layers in the form of mesoderm. Despite some differences in developmental patterns, comparative embryological studies overwhelmingly support the concept of Darwinian evolution. The species that follow the same pathway for several steps after the gastrula produce adult forms that are more similar than two species whose pathways diverge after that stage. The different developmental patterns are thought to be indications of different lines of evolution. Another feature of embryological development that serves to link all multicellular animals is the embryological source of organ systems. Regardless of the way in which the developing animals are programmed for further differentiation of mesoderm, they exhibit similar adult structures derived from the two primary germ layers. The outer covering of all multicellular animals, be it skin, scales, or gelatinous material, is derived from ectoderm. The universal features of ectodermally and endodermally derived tissues also indicate the presence of a common ancestral type early in evolutionary history. Besides these basic embryological similarities in all multicellular animals, there exist certain embryological aspects which may compel one to believe in Darwinian evolution. For instance, it is most commonly observed that the embryos of higher animals repeat many of the stages through which embryos of higher animals have passed. This has been referred to as recapitulation. This concept, as originally used by von Baer, 1792-1876, indicated that some of the developmental stages of an organism are similar to some of the developmental stages of its ancestors. Unfortunately, however, E. Haeckel in 1866 modified the concept into a biogenetic law, which stated that ontogeny recapitulates phylogeny, i.e., in its development, the individual passes through stages like the adult stages of its ancestors. The Haeckelian idea is wrong in the eyes of modern embryologists, yet it has stimulated research in embryology and focused attention on the general resemblance between M at W. Fish, salamander, tortoise, chick, pig, cow, rabbit, man. Figure 4.9. Comparison of three embryonic stages from fish to man. Embryonic development and the evolutionary processes. According to biogenetic law of Haeckel, the fertilized egg can be compared to the hypothetical single-celled flagellate ancestor of all animals, and the blastula can be compared to a colonial protozoan or to some hypothetic blastula-like animal which has been postulated to be the ancestor of all metazoa. Haeckel believed that the ancestor of coelenteroids and all the higher animals were a gastrula-like organism with two layers of cells and a central cavity connected by a blastopore to the outside. Modern scientists prefer von Bayer's recapitulation concept than biogenetic law of Haeckel. There are numerous examples of recapitulation phenomenon during embryonic development, but one of the best is afforded by a comparison of different vertebrate embryos at comparable stages in development, see figure 4.9. During the early embryonic stage in the series, all the embryos look very much alike. All have similar pharyngeal arches and pharyngeal clefts. At somewhat later stage in the series, the limb bud primordia of fore and hind limbs are forming in all embryos in a similar way and all of them have embryonic tails. The embryos of the lizard, chick, opossum, monkey, and man have strong resemblances, yet those of the fish and salamander are beginning to assume recognizable forms. At this stage, gills have formed from the tissues lining the gill clefts of both the fish and salamander. Later, each embryo has developed features that indicate fairly clearly its definitive nature. Further, in the development of any mammalian embryo, the heart is a four-chambered in-series structure, as it is in fish embryo. Then it develops partitions of the auricle atria, similar to those of amphibian embryos, followed by ventricular division that is incomplete for a period, as it is in the embryos of reptiles. A similar example of recapitulation is found in mammalian embryos in the formation of pronephric, mesonephric and metanephric kidneys. The existence of a common trochophore larval stage in the development of many mollusks and many annelids is also typical of this process. Plants provide a few examples of recapitulation. One good example is found in the acacia tree. This tree has well-developed compound leaves, yet the seedling has simple leaves like those found in all stages of development of its ancestors. Further, it is generally believed that mosses and ferns are more highly evolved than algae. Protonema of mosses, which is a green, filamentous structure that originates from an asexual spore, resembles certain green algae. This provides a clue to their evolutionary relationship. Both bryophytes and pteridophytes have ciliated sperms and require water for fertilization. Gymnosperms do not need water for fertilization, but cycads and ginkgo, the primitive gymnosperms, have ciliated sperms like the pteridophytes. This suggests that gymnosperms have descended from pteridophyte-like ancestors. Genetic basis of recapitulation. Our increasing understanding of physiological genetics provides us with an explanation of the phenomenon of recapitulation. All chordates have in common a certain number of genes which regulate the processes of early development. As mammalian ancestors evolved from fish through amphibian and reptilian stages, they accumulated mutations for new characteristics but kept some of the original, fish, genes, which still control early development. Later in development, the genes which the human or any other mammal shares with amphibians influences the course of development so that the embryo resembles a frog embryo. Subsequently, some of the genes which have in common with reptiles come into control. Only after this stage, most of the peculiarly mammalian genes exert their influence, and these are followed by the action of genes human has in common with other primates. The anthropoid apes, which have the most immediate ancestors in common with man, have the most genes in common with him and their development is identical with him, human, except for some fine details. In general, during development the general characteristics that distinguish phyla and classes appear before the special characteristics that distinguish genera and, finally, species. Within each phylum, the higher forms pass through a sequence of developmental stages which are similar to those of lower forms but achieve a different final form by adding changes at the end of the original sequence and by altering certain of the earlier embryonic stages they share with the lower forms. Villi et al., 1973. Evidences from comparative physiology and biochemistry. Basically, evolution is biochemical phenomenon and it is, therefore, natural that physiology and biochemistry have given some of the following most important and dependable evidences to support the idea of evolution. 1. Protoplasm chemise tri. Biochemical analysis of the living matter in the protoplasm, which is considered as the physical basis of life, suggests that protoplasm from a variety of sources, i.e., bacteria, blue-green algae, plants and animals, has the same biochemical constitution. It mainly consists of substances like proteins, lipids, carbohydrates, water, etc. This would suggest that during evolution the most fundamental property of living things has remained intact, while variations in certain essential respects produce the variability according to the needs of differential forms. 2. Chromosome chemise tri. Like protoplasm, another remarkable similarity at biochemical level is found in the chemistry of chromosomes. The chromosomes of all living organisms basically consist of nucleic acids, DNA and RNA, and proteins, histones and protamines. The molecules of these chemical substances remain arranged in all chromosomes in an almost identical fashion. Such a uniformity in the composition of chromosome again suggests a common origin of most living beings. 3. Enzyme similarities. A large number of animals and plants contain identical enzymes. Several enzymes found in the digestive tract are common in a variety of animals. For example, trypsin and amylase are found from sponges to mammals. A number of enzymes used in photosynthesis are common in a variety of green plants. Such common enzymes and consequently a common mechanism of process of photosynthesis suggest a common ancestry of green plants. 4. Hormonal similarities. Like enzymes, hormonal similarities are also as found in all vertebrates. For example, thyroid hormone sedum Eg_L_. U. Mone is commonly found in all vertebrates and this E hormone from one class of organism can be when a foreign protein is inoculated into the blood of an animal, the latter produces a complex protein compound against that foreign protein inoculated. These compounds are familiarly known as all antibodies and the foreign inoculated protein is known as antigen. When a reaction occurs B. Dog serum equals. Figure 4.10. Principle of precipitin test applied to investigation of animal relationship. Tween antibody and antigen, a soft white precipitate will be formed. The strength of precipitate depends upon the concentration of antigen. The precipitate is the precipitin and the test is precipitation test. One of the remarkable features of this test is that the antibodies formed against one antigen can also react with antigens of other source, provided the latter is chemically similar to the first antigen. Antibodies containing serum is known as antiserum. Antiserum of antigen of one animal can be tested with antigens of other animals in order to show their relationships. The test can be interpreted that if precipitate results with more diluted antigen of one animal against the test animal, then the former is more closely related to the latter. If precipitate results with less diluted antigen of that animal, then it is distantly related to the test animal. Such precipitin tests have been conducted to resolve the disputed relationships of organisms in recent years. Of scores of examples, here we give 2 illustrations. Till recently, I disbelieved that whales have relationship with fishes. It is because almost all of their anatomy are so strongly modified to aquatic fish-like life. Only few anatomical clues to show their relationships to other mammals remained. However, comparative serology of whales with other mammalian groups indicates that their serum proteins are most like those of the even-toed hoofed order Artiodactyla mammals. This might suggest that whales sprang from primitive artiodactyl stock. The same serological tests when performed in slightly modified form among the members of primates, man, and anthropoid. Ape, an Old World monkey, a TR New World monkey and a LeSheep serum G.F. UIMER, the amount of precipitate of serum proteins would O decrease in descending order, sheep serum I rabbit serum that is, the anthropoid ape is U ilgolution. More closely related to man I than other organisms. Even. Among anthropoid primates, L. agtref LDFPE Goa, GTGM-LSQG tests done according to I sheep serum placed Australoni technique, goat serum and percentin I in each tube, Goodman, 1962, 1967, reserial dilutions revealed that the serum proteins of chimpanzee are more alike to man's serum proteins than the I. Serum prote. NS of Asia's tick apes, SERB IEALFDSIELRUUT ions. Gorilla and baboon. Closely related. Cows, sheep, pipe. Goats, deer and antelopes coney. Relationship among the modern birds than among the mammals. For all of the several hundred species of birds tested give strong and immediate reactions with serum containing antibodies for chicken serum. From other tests it was concluded that birds are more closely related to the crocodile line of reptiles than to the snake lizard line, which corroborates the paleontological evidence. Similar tests of the sera of crustaceans, insects and mollusks have shown that forms regarded as being closely related from morphologic and paleontologic evidence also show similarities in their serum proteins. Figure 4.11. Principle of precipitin test employing serial dilutions of antigen and the interfacial reaction. 6. Amino acid sequence analyses. Molecular biological investigations of the sequence of amino acids in the O and 8 chains of hemoglobins from different species have revealed great similarities, of course, and specific differences, the pattern of which demonstrates the order in which the underlying mutations, the changes in nucleotide base pairs, must have occurred in evolution. The evolutionary relationships inferred from these studies agree completely with those based on morphologic studies. Analyses of the amino acid sequence in the protein portion of the cytochrome enzyme provide further concurring evolutionary relationships. Further, the pattern and rates of reactions of lactate dehydrogenase and certain other enzymes with the normal pyridine nucleotide coenzyme, NAD, and with analogs of NAD can be used to demonstrate evolutionary relationships. 7. Excretory product analyses. An analysis of the urinary wastes of different species provides the evidence of evolutionary relationship. The kind of nitrogenous excretory waste depends upon the particular kinds of enzymes present, and the enzymes are determined by genes, which have been selected in the course of evolution. The waste products of the metabolism of purines, adenine and guanine are excreted by man and other primates as uric acid, by other mammals as allantoin, by amphibians and most fishes as urea, and by most invertebrates as ammonia. Vertebrate evolution has been marked by the successive loss of enzymes for the stepwise degradation of uric acid. J. Needham made the interesting observation that the chick embryo in the early stages of development excretes ammonia, later it excretes urea and finally it excretes uric acid. The enzyme uricase, which catalyzes the first step in the degradation of uric acid, is present in the early chick embryo but disappears in the later stages of development. The adult frog excretes urea, but its tadpole larva excretes ammonia. These biochemical examples are the repetition of the principle of recapitulation. 8. Phosphagens. The phosphagens play a key role in muscle contraction and are the sources of energy for the resynthesis of ATP, once they are broken down. In the muscles of most vertebrates, phosphagen is always a specific compound called creatine phosphate, while in most invertebrates it is arginine phosphate. Hemichordates, the most primitive chordates, have both the phosphagens, the creatine phosphate as well as arginine phosphate. Such a situation is also found in echinoderms, and on morphological grounds echinoderms have been considered close to the ancestor of chordates. Evidences from comparative cyioiagy. Another type of evidence that indicates that all forms of life are related comes from the cellular level. The very fact that the cell is the unit of structure for all living organisms, except viruses, is thought to reflect the basic relationship among living forms. This relationship is even further emphasized by the fact that it has been possible for biologists to construct a picture of the generalized cell from which all other types can be inferred. Moreover, all cells that have been examined thus far have a DNA RNA protein information and communication system. All forms contain membranes that are made up of double-layered lipoproteins. All cells, except few bacteria, utilize the glycolytic pathway. Most bacterial forms, and all uni and multicellular organisms, have a Krebs cycle and an electron transport system. All are based on ATP as an energy donor. Certainly these factors provide an overwhelming demonstration of the interrelatedness of biological forms. Evidences from genetic S. Genetics, the science of heredity, deals with the variability of plants and animals. Hereditary variations provide the raw material of evolution. There are mainly two sources of hereditary variations, namely recombination and mutation. While recombinations after hybridization yield new combinations, mutations will create new genetic material which never existed earlier. For the past several thousand years man has been selecting and breeding, i.e., hybridizing, animals and plants for his own uses, and a great many varieties, adapted for different purposes, have been established. These results of artificial selection provide striking models of what may be accomplished by natural selection. All of our breeds of dogs have descended from one or perhaps a very few species of wild dog or wolf, yet they vary so much in color, size and body proportions that if they occurred in the wild they would undoubtedly be considered separate species. They are all interfertile and are known to come from common ancestors, so they are regarded as varieties of a single species. A comparable range of varieties has been produced by artificial selection in cats, chickens, sheep, cattle and horses. Plant breeders have established by selective breeding a tremendous variety of plants. From the cliff cabbage, which still grows wild in Europe, have come cultivated cabbage, cauliflower, kohlrabi, Brussels, sprouts, broccoli and kale. Further, cytogeneticists have been able to trace the ancestry of certain modern plants by a combination of cytologic techniques in which the morphology of the chromosomes is compared and by breeding techniques which compare the kinds of genes and their order in particular chromosomes in a series of plants. In this way, the present cultivated tobacco plant, Nicotiana tabacum, was shown to have arisen from two species of wild tobacco, and corn was traced to teosinte, a grass-like plant which grows wild in the Andes and Mexico. Moreover, the cytologic details of the structure of the giant chromosomes of the salivary glands of fruit flies have been of prime importance in unrevealing the evolutionary history of many species of Drosophila. At B. Mutation seen in Zinnia petals, A, and Drosophila B. Mutations have also played a very significant role in evolution. Different kinds of mutations, namely gross mutation, due to variation in chromosome number, polyploidy and aneuploidy, and point mutations, see chapter of mutation, introduce different kinds of variations in plants and animals and consequently result in speciation. Plant breeders have employed induced polyploidy methods in producing numerous economically important varieties of plants. Geneticists have produced many new strains of microorganisms, plants and animals Drosophila, by artificially inducing point mutations in them. Thus, the artificial selection methods due to recombination or polyploidy and induced methods of mutations have suggested the fundamental processes which may be involved in organic evolution. Evidences from biogeographical relations. Biogeography deals with the manner in which plants and animals are distributed over our planet. On the basis of similarities in the existing fauna, found in different regions of the Earth, the following 6 biogeographical regions have been distinguished. A. Nearctic, North America down to the Mexican Plateau. B. Palearctic, Asia north of the Himalayas, Europe and Africa, north of Sahara Desert. C. Neotropical, Central South of America. D. Oriental, Asia, south of the Himalayas. E. Ethiopian, Africa, south of the Himalayas. F. Australian, Australia and the associated islands. Any Onua familiar with biogeography of South American and African regions, immediately be convinced by the fact that similar habitats are populated with similar animals. These two are extensive tropical regions and crossed by the equator. They both have similar habitats in that, have lowland jungles, extensive river systems and mountain regions. Both regions extend southward to temperate zones. Yet, surprisingly, they have dissimilarities in fauna more greatly than similarities. The characteristic fauna of African Pludeslians, elephants, rhinoceroses, hippopotami, many kinds of antelopes, giraffes, zebras, hyenas, lemurs, baboons, monkeys with narrow noses and non-prehensile-tailed chimpanzees and gorillas. Alpacas. In South America, monkeys occur with broad noses, tapirs, odd-toed hoofed mammals, rodents like capybara, agouti, chinchilla and paca, mountain lions like panthers, ocelots jaguars of cat family, well-known uamas, guanacos, vicuñas, and alpacas, armadillos, many opossums, giant anteaters, raccoons, spectacled bears and sloths and many others. Thus, if both Africa and America have similar varying habitats, then how could it have happened to them to possess different fauna instead of similar fauna? We cannot simply satisfy with such an answer that suitability of habitat is important. Because we do find similarities in fauna of both regions. They both have such widely ranging animals, bats, rats, SQ9 etc. The other alternative answer might be the accessibility of these two regions to the animals found in them. Let us examine this point here. The flightless kiwi is found only in New Zealand, as were the giant flightless birds called moas. Moas are now extinct. Moas and kiwis evolved from flying birds that vicuñas. More briefly, Simpson has established a relationship, faunal stratification, between the separateness of animals and their period of length of appearance in America. Based on it, he concludes that armadillos and sloths are the oldest inhabitants of America since early Cenozoic times. They have not appeared in other part of the world. Monkeys and field mice have formed part of American fauna during mid and late Cenozoic times, while the rest of the fauna have independently evolved. These facts have been ascertained by fossil record. The record reveals that during long periods American forms have no contact with those on other continents. Occasionally animals might have reached the continent by island hopping across the intervening sea. This type of dispersal is best seen in fauna on continental islands. For the most part the isthmus of Panama that links colonized islands of New Zealand millions of years ago. Early in the Tertiary period, when mammals were still all fairly small, moas did in fact dominate. Moas died out quite recently because of overhunting. America to America was submerged and thereby the two continents are isolated. When once geographical isolation takes place, local fauna along with invaders might evolve into distinct species in many different times and have become unique to America. O equals X, L. Y underscore I Greenland or E equals U. I, U I. Dot. L. O. N. Thus, this is the only possibility for the same habitats in different parts of Earth have been populated by different animals and plants. This fact suggests that the animals today we see in different biogeographical regions have descended from their predecessors with different structures, and have migrated from their place of origin to their new areas, but have failed to return to their place of origin, because it is separated geologically. Continental islands. These islands are located on continental shelves of continents. These islands sometimes are connected to mainland when ocean water recedes or the level of island rises. The fauna and flora of continental islands characteristically resemble those of continents to which they were formerly joined. One interesting feature is the presence of amphibians and mammals on continental islands. Island hopping in these animals is less likely and, hence, they might have arrived on these islands through land connections. Thus, these evidences have established that biological evolution is a fact but not a dogma. Consequently, the biologist's conviction that, through a series of changes resulting from natural selective processes, life came to the state known today, is of such magnitude that the entire science of biology has been oriented according to the evolutionary doctrine. Organisms have been reclassified according to proposed evolutionary relationships. Geneticists interpret their results as possible mechanisms of evolution or sources of variation. So powerful is this idea today that only a text organization based on evolutionary doctrine would truly represent the science of biology. Theories of organic evolution. Lamarckism, Darwinism, modern synthetic theory, germplasm theory and mutation theory. Different forms of life, special creation and evolution. Many evidences have been presented in previous chapters to support the concept of evolution, and almost without exception modern biologists are convinced of evolution. Up till now, the following convincing theories have been forwarded to explain the mechanism of evolution. The first of these theories, which is now only of historical significance, was that of Jean-Baptiste de Lamarck, 1744-1829. 1. Theory of inheritance of ACQUIRED characters, IAMARCKISM. The theory of inheritance of acquired characters states that modifications which the organism acquires in adaptation to the environments which it meets during its lifetime are automatically handed down to its descendants, and so become part of heredity. This theory was propounded by a renowned French naturalist, Lamarck, shortly after the occurrence of French TWO explanations have been offered for the origin of the competition between animals for food, space, light and moisture as an aspect of struggle for existence. Revolution. Lamarck spent the early part of his life as a botanist. Then at the age of 50 he turned his attention to zoology, particularly to the study of invertebrates. The terms invertebrate and biology have been coined by Lamarck. As a result of his systematic studies he became convinced that species were not constant but rather were derived from pre-existing species. This idea was in total conflict with the view of the period, that of fixity of species. As a result Lamarck's views were challenged by most of the biologists of that time, particularly by Georges Cuvier. In 1809, Lamarck published Philosophie zoologique, which included his theory explaining the changes that occur in the formation of new types. Although his views on evolutionary mechanism are outmoded now, he still occupies a very important place in the history of evolutionary thought. He was the first evolutionist to conclude that evolution is a general fact covering all forms of life. His evolutionary ideas can be discussed in brief as follows. 1. Internal forces of life tend to increase the size of the organism. New structures appear because an inner want of the organism, i.e., the internal forces of life tend to increase continuously the size of an organism and its component parts. 2. Direct environmental effect over living organisms. The organs of an animal became modified in appropriate fashion in direct response to a changing environment. 3. Use and disuse. The various organs became greatly improved through use or reduced to vestiges through disuse. 4. Inheritance of acquired characteristics. Such bodily modifications, in some manner, could be transferred and impressed on the germ cells to affect future generation. Thus, inheritance was viewed by Lamarck simply as the direct transmission of those superficial bodily changes that arose within the lifetime of the individual owing to use or disuse. Volpe, 1985. Thus, Lamarck believed that organic changes seen in animals were resulted by the influence of environment on the gradual changes of species due to their tendency to become more and more perfect. According to him, when an animal's environment changes, its needs change, and this leads to special demands on certain organs. Organs used more extensively would enlarge and become more efficient. Conversely, an organ or organs, no longer used, would degenerate and atrophy. He postulated that such changed characteristics, acquired traits, would be transmitted to the offspring. Examples of Lamarck's M. 1. The deer-like ancestor of giraffe lived in places, Africa, where the ground was almost invariably parched and without grass. Obliged to browse upon trees, it was continually forced to stretch upwards. This habit maintained over long periods of time by every individual of the race had resulted in the forelimbs becoming longer than the hind ones, and neck so elongated that a modern giraffe can raise his head to a height of 18 feet without taking his forelimbs off the ground. 2. Ducks and other as aquatic birds in Bededwa registered tours from land in search of equals, enough food, because figure 5.1. Evolution of giraffe according to Lamarck. Food was scarce on land. And these birds did not had power to fly. In water, the duck would stretch its toes apart to give more push during swimming. This new characteristic would be inherited, and the subsequent generation of duck would upon stretching their toes form a more defined web. Each generation would do the same until the webbed foot seen on ducks today was fully formed. This would then be passed on from generation to generation, essentially unchanged once the perfected state was attained. 3. Flat fishes, deep sea fishes present at the bottom of sea where there is no sunlight, led an inactive life, lying on one side of the body. The eye of that side, lying towards bottom, migrated towards upper side and, thus, both eyes are on one side of the body. 4. The whales lost their hind limbs as the consequence of the inherited effect of disuse. 5. The wading birds, e.g., jacana, developed its long legs through generations of sustained stretching to keep the body above the water level. 6. Snakes have elongated body accompanied by loss of limbs. The continuous creeping through holes and crevices made limbs continuously useless for locomotion with the result that limbs become completely lost in snakes. 7. Eyes are reduced in moles since they live underground. In cave animals also, eyes might become functionless purple heron. According to Lamarck's. And might even disappear. Theory, by trying to keep its belly out of the water a wading bird purple heron acquires the habit of stretching and elongating its legs. 8. For plants Lamarck accepted the theory of his compatriot Saint Hilaire, who developed the notion that plant form is shaped by the combined effects of the environment. There are other effects such as vestigial organs in living animals due to disuse. Claws in carnivora, sensitive skin and tactile points on the ventral side of the body and callosities of palm in hard workers, exemplifying Lamarckian theory. Significance of Lamarckism. Lamarckian theory was simple and it had some appeal, as it provided a way in which changes in organisms could come about. It was the first completely comprehensive mechanistic theory that was offered. Furthermore, it was the theory that lent itself to predictions and, therefore, to testing. Thus, Lamarckian. Trofim Lyenko, 1898-1976, Soviet geneticist, favored Lamarckian ideas, since they agreed with communist ideology and he rose to power under Stalin. Lysenko banished Mendelian geneticists and dominated Soviet genetics for many years. Eventually, however, the evidence against Lamarckian inheritance was so strong that ideology had to give way to science. Lysenko was discredited and forced to resign. Theory enjoyed popular acceptance for near about 70 years, because it was exemplified by many common examples. Most persons know that exercise results in larger muscles. Critical analysis of Lamarck's propositions. Lamarck defended his evolutionary theory vigorously until his death. For it, he suffered both social and scientific ostracism, but he had the courage of his convictions. However, he was criticized for the following reasons by the contemporary scientists during his lifetime and afterwards. 1. The first proposition of Lamarck suggests the tendency to increase in size. While the evolutionary trend in a certain groups of organisms may be associated with the increase in size, there are many cases where evolution proceeded not only without any increase in size but rather through a reduction in size. Many plants contradict this Lamarckian principle by showing such a reduction in size during their evolution. Many ferns and conifers which became extinct were gigantic trees and the more highly evolved flowering plants are really much smaller in size. 2. The second Lamarckian principle that new organs result from new needs is quite manifestly false. In the case of plants, Lamarck believed that the environment acted directly upon the plant, causing the production of such new characters as might adapt the plant to its environment. In the case of animals, he believed that the environment acted through the nervous systems. In other words, the desire of the animal leads to the formation of new structures. In its crudest form, this would mean that the man who mused, "Birds can fly, so why can't I?" should have sprouted wings and taken to the air. 3. The third Lamarckian principle that organs will develop due to use and degenerate due to disuse may be correct as far as growth of an organ within the lifetime of an individual is concerned. For example, it is a commonly observed fact that if muscles are put to use, these would develop. However, this principle is meaningful only when it is studied in relation to the following fourth principle. 4. The fourth and final proposition of Lamarck was that the inheritance of characters acquired during the lifetime of the individual. This principle has been tested by many biologists who have devised many types of experiments for it and have found it entirely incorrect. Certain experiments which have discredited it are the following. 1. The noted German scientist August Weismann was the first person who for the first time made a definite distinction between heritable changes and those which cannot be inherited. In 1890, he performed some experiments to test if characters may disappear due to disuse. This he did by cutting the tails mutilation of white mice for more than 20 generations to see if this has any effect on tail length. The measuring of tail length of the offsprings of 20 successive generations revealed that on average, the tails were not shorter. It means that, acquired August Weismann, 1834-1914. Character cut tail was not inherited. 2. Castle and Phillips performed transplantation experiments to show that environment has no effect on heredity. In one of the experiments they transplanted the ovary of a black female guinea pig into the body of white female guinea pig and the recipient female was mated with a white male guinea pig. They found that all the individuals from this pair were black. This shows that the environment does not affect the heredity as has been suggested by Lamarck. 3. Loeb artificially fertilized the eggs of sea urchin by certain chemical stimuli and these parthenogenetically fertilized eggs produced the generations, the members of which possessed no parthenogenesis. 4. Boring of ears and nostrils in Indian women has been continued as the tradition from centuries but their offsprings do not show any trace of holes in ears and nostrils. 5. The Chinese bound the feet of their women for many generations, yet this has not resulted in any modification of the feet of present-day Chinese women. 6. Jewish boys, and also Muslim boys, have been circumcised for thousands of years, but this has not resulted in a tendency toward the reduction of the prepuce in them. All the aforesaid cases and experiments clearly showed that Lamarckian theory is not tenable. Neo-Lamarckism. M. The evolutionists who support the Lamarckian doctrine of inheritance of acquired characters come under the heading of Neo-Lamarckian. Among the Neo-Lamarckians' notable supporters are Cope (1840–1897), Giard (1846–1908), Packard, Spencer, and McBride tried to modify Lamarckism in order to make it acceptable. These Neo-Lamarckians considered that adaptation is universal. It arises as a result of causal relationship of structure, function, and environment. Changed environmental conditions alter habits of organisms. Hence, in response to new habits, organisms acquire new structures in place of old structures. Consequently, variations among animals have become distinct. These variations have finally become ingrained in the heredity of the race. The kind of argument is, in fact, a modified version of Lamarckian principles, because it has omitted Lamarck's view that of a general perfecting tendency in evolution. It stressed direct action of environment on organic structure. For example, according to Neo-Lamarckians, the development of fur on skin by animals as protection against cold weather is the consequence of changed environment from warmer to colder state only. But if the environment reverts to normal state, the fur would also disappear. To account it, Neo-Lamarckism included also the effects of use and disuse. Based on this, Neo-Lamarckians have rejected natural selection as the sole mechanism of evolution. On the other hand, they believed in the interplay of structure, function, and environment as the whole truth of evolution. However, no evolutionist of today is adherent to Neo-Lamarckism. The following are the evidences of transmission of acquired characters. 1. McDougall conducted learning experiments in rats, and from the results obtained he tried to suggest that learning as an acquired character can be inherited. During his experiments, the rats were dropped into a tank of water from which there were two exits, one lighted and one dark, but not always the same one. A rat leaving by the lighted exit received an electric shock, while one leaving by the dark exit received no shock. Thus, the number of trials required for a particular rat to learn always to select the dark exit constituted a measure of the speed of learning. These rats were then bred and their descendants were similarly studied. It appeared that the speed of learning increased from generation to generation, and so, McDougall concluded that learning, an acquired trait par excellence, is inherited. Some serious criticisms have been raised against McDougall's experiments. It is suggested that genetic constitution of rats was not controlled. Moreover, in the progeny of control rats not subjected to these learning tests, the learning habit was found to change the same way as in the treatments. So, it was said that some unanalyzed changes in the technique may be responsible for the recorded increase in the speed of learning. Lastly, the most strange aspect about McDougall's experiment is that when the same experiments, when repeated in other laboratories, never gave similar results. 2. F.B. Summer reared the white mice at 20 degrees Celsius to 30 degrees Celsius, because of which they developed longer bodies, tail and feet. When these mice were bred at lower temperatures, their offspring showed their normal proportions of size. 3. Lindsay subjected certain cold-blooded animals, warm-blooded animals and plants to unusual environmental conditions and found that environment affects certain changes in these organisms, and these acquired modifications were inherited up to some extent in their offsprings. 4. Gubber and Smith induced the hereditary changes in the eyes of fetuses of rabbit by simply destroying lens of living female with a needle in situ. The anti-lens serum has been produced in the blood of these animals. 5. Griffith and Detlefsen reared rats in cages placed on rotating table for several months. Consequently, they became adapted to the rotating condition to such an extent that when rotation was stopped, they showed signs of nystagmus, dizziness, and other physiological conditions. This condition was inherited for several generations. 6. Various acquired diseases described by Brown-Sequard are inherited from generation to generation. For example, exophthalmia was caused in parents by injuring in brain the restiform body. This disease was, later on, inherited to several generations. The other diseases such as hematoma and dry gangrene are produced by injury to the restiform body near the rib of calamus. Later, these were found to be inherited. 7. Kammerer worked on the tailed amphibian Proteus anguinus, which lives in complete darkness in the water of underground cave. It was blind and colorless. He brought Proteus in daylight, due to which it became colored, brown and black, which passed on to its progeny. Eyes of Proteus also developed normally in daylight. This showed that any change in environmental conditions induced changes in the animals, and the acquired characters were inherited. Further, Winterburt 1962 has tried, with some success, to give Lamarckism a chemical basis by involving the capacity of every living being to react to environmental changes and aggressions. According to Winterburt, nothing is acquired by the living organism that is not the response of internal factors to an outside influence or ethological change. Living means reacting, never undergoing. Above all, it means not waiting around for a fortunate chance occurrence to save the situation. Grass, 1977. 2. Theory of Naturalselection. Darwinism. More than a century ago, in 1859, Charles Robert Darwin (1809–1822) gave the biological world the master key that unlocked all previous perplexities regarding the mechanism of organic evolution. His natural selection theory can be compared only with such revolutionary ideas as Newton's law of gravitation and Einstein's theory of relativity. Volpe, 1985. The concept of natural selection was explained clearly and convincingly by Darwin in his masterpiece, The Origin of Species. The full title of the book was On the Origin of Species by Means of Natural Selection, or the Preservation of Favored Races in the Struggle for Life. This epoch-making book was the fruition of more than 20 years of meticulous accumulation and analysis of facts. The first edition of the book, some 1,250 copies, was sold out on the very day it appeared, November 24, 1859. This book had opened a Pandora's box. It was immediately both acidly attacked and effusively praised. Charles Robert Darwin was born on February 12, 1809 in Shrewsbury, England. His personality was not the type that one associates with a successful man, let alone a great one. While his family held a distinguished reputation, his grandfather Erasmus Darwin was a noted poet-scientist, his father Robert was an eminent physician, and his mother was a Wedgwood. The family of pottery fame. Charles was a singularly undistinguished individual whose contemporaries viewed him as just a normal, gentle, cautious, morally upright English lad. After completing his early education at Shrewsbury, Charles Darwin was sent at the age of 15 to study medicine at the University of Edinburgh. His medical interests soon dissipated, and he became something of an academic rover, studying a little science, but finding it too formal for his tastes. His father then suggested him to become a clergyman, which appealed him. So, he got transferred, after 2 years, to Christ's College, Cambridge University, to study theology. There too he spent his energy in card playing and drinking. He was obviously not ready for clerical commitments. He did manage, however, to find some constructive interest in natural history and in collection of natural objects of all kinds. His friendship with Professor John Stevens Henslow, botanist at Cambridge, stimulated an interest in botany, while his exposure to the geologist Sedgwick stimulated an interest in geological explorations. Through the recommendation of Dr. Henslow, Darwin accepted a most inauspicious position as a naturalist without pay aboard the HMS Beagle. The ship was to spend 5 years in exploration, mostly of South America. Darwin was to provide a collection of material representing the natural composition of the areas visited. The ship Beagle left Plymouth on December 27, 1831 and visited many islands of the Atlantic Ocean, some coasts of South America and some islands of South Pacific, of which Galapagos Islands are the most important. During his 5 years, from 1831 to 1836, on the Beagle, Darwin recorded almost everything he observed and sent an enormous amount of material back to England. Upon his return home in 1836, he spent 2 years writing a book of his experiences, The Voyage of the Beagle. In 1838, he got married and later had 2 daughters and 5 sons. From 1838 to 1841, Darwin acted as the Secretary of Geological Society, where he came in contact with an eminent geologist, Charles Lyell. It was not until 1844 that Darwin developed his idea of natural selection in an essay, but not for publication. He showed the manuscript to Lyell, who encouraged him to prepare a book. Darwin still took no steps toward publishing his views. Perhaps he was well aware of how Lamarck's earlier theory had been received and wanted all possible supporting evidence before publishing his own new theory. It appears that Darwin might not have prepared his famous volume had not a fellow naturalist in the Dutch East Indies, Alfred Russel Wallace 1823, 1913. Eel. Independently conceived of the idea of natural selection. Wallace had traveled widely in tropical South America and southeastern Asia, for studying the flora and fauna of these regions. Wallace also inspired by reading Malthus's essay, and the idea of natural selection came to him in a flash of insight during a sudden fit of malarial fever. This happened in February, 1858, when he was working in the island of Ternate in Indonesia. In June of 1858, Wallace sent Darwin a short essay, on the tendency of varieties to depart indefinitely from the original type, and asked him if he thought of sufficient interest to present it to the Linnaean Society. We can easily imagine Darwin's amazement upon receiving Wallace's essay. Upon the insistence of the geologist Charles Lyell and the botanist Joseph Hooker, Darwin prepared an abstract of his conclusions for joint publication with Wallace's essay. Wallace's essay and a portion of Darwin's manuscript, each containing remarkably similar views, were read simultaneously before the Linnaean Society in London on July 1, 1858. The joint reading of the papers stirred little interest. Darwin then labored for 8 months to compress his voluminous notes into a single book, which he modestly called, only an abstract. Wallace shares with Darwin the honor of establishing the mechanism by which evolution is brought about, but it was the monumental The Origin of Species with its impressive weight of evidence and argument that left its mark on mankind. The fact that this book, written by Darwin alone, exposed the public at large to the theory of natural selection, and that Wallace was still in Indonesia at the time of the controversy and could not champion Darwin's defense by adding his own views, brought the public to associate only Darwin with this theory. Thus the theory is called the Darwinian, rather than the Darwin-Wallace theory of evolution. Darwin died on April 19, 1882, when he was 73 years old. During his life, he wrote numerous books, journals, etc. Some of them can be listed in Table 5.1. Some publications of Charles Darwin. Title of book, year of publication. 1. Journal of Researches 1839. 2. The Structure and Distribution of Coral Reefs 1844. 3. Geological Observations on South America 1846. 4. A Monograph on Cirripedia Barrancles, Living and Fossil, 4 volumes 1851, 1854. 5. Origin of Species 1859. 6. The Fertilization of Orchids 1862. 7. The Variation of Plants and Animals under Domestication 1868. 8. The Descent of Man 1871. 9. The Expression of the Emotions in Men and Animals 1872. 10. Insectivorous plants 1875. 11. The effects of cross and self-fertilization in the vegetable kingdom 1876. 12. Different forms of flowers on plants of the same species 1877. 13. The power of movement in plants 1880. 14. The formation of vegetable mold through the action of worms 1881. Facts that influenced Darwin's thoughts. During the period in which Darwin massed his evidence and developed his natural selection theory, many things affected his thinking, of which 3, in particular, deserve mention here. 1. During his voyage, Darwin took only a few books on board. One of them was the newly published first volume of Charles Lyell's Principles of Geology, a parting gift from his Cambridge mentor, John Henslow. In this book, Lyell challenged the prevailing belief that the Earth has been created by a divine plan nearly 6,000 years ago. On the contrary, the Earth's age could be measured in hundreds of millions of years. Lyell asserted that the Earth's mountains, valleys, rivers, and coastlines were shaped not by nose flood but by the ordinary action of the rains, the winds, earthquakes, volcanoes, and other natural forces. Darwin was impressed by Lyell's emphasis on the great antiquity of the Earth's rocks, and gradually came to conclusion that the characteristics of organisms as well as the face of the Earth could change over a vast span of time. The living and extinct organisms that Darwin observed in the flat plains of the Argentine Pampas, Pampas are vast grassy treeless plains in South America, and the Galapagos Islands sowed the seeds of Darwin's views on evolution. From old riverbeds in the Argentine Pampas, he dug up bony remains, fossils, of extinct mammals such as Toxodon, Macrauchenia, Pyrotherium and Thotherium, figure 5.3. The presence of Thotherium testified that a horse had been among the ancient inhabitants of the continent. It was the Spanish settlers who reintroduced the modern horse, Equus, to the continent of South America in the 16th century. Darwin wondered that a native horse should have lived and disappeared in South America. This was one of the first indications that species gradually became modified with time, and that not all species survived through the ages. Further, when Darwin collected the remains of giant armadillos and sloths on an Argentine pampa, he revealed the fact that, although they clearly belonged to extinct forms, they were constructed on the same basic plan as the small living armadillos and sloths of the same region. This observation initiated him to think of the fossil sequence of a given animal species through the ages and causes of extinction. Moreover, what Darwin had appreciated was that living species have ancestors. On traveling from the north to the south of South America, Darwin observed that one species was replaced by similar, but slightly different, species. In the southern part of Argentina, Darwin caught a rare species of ostrich that was smaller and differently colored from the more northern common American ostrich, Rhea americanus. Moreover, facts that influenced Darwin's thoughts. During the period in which Darwin massed his evidence and developed his natural selection theory, many things affected his thinking, of which 3, in particular, deserve mention here. 1. During his voyage, Darwin took only a few books on board. One of them was the newly published first volume of Charles Lyell's Principles of Geology, a parting gift from his Cambridge mentor, John Henslow. In this book, Lyell challenged the prevailing belief that the Earth has been created by a divine plan nearly 6,000 years ago. On the contrary, the Earth's age could be measured in hundreds of millions of years. Lyell asserted that the Earth's mountains, valleys, rivers, and coastlines were shaped not by nose flood but by the ordinary action of the rains, the winds, earthquakes, volcanoes, and other natural forces. Darwin was impressed by Lyell's emphasis on the great antiquity of the Earth's rocks, and gradually came to conclusion that the characteristics of organisms as well as the face of the Earth could change over a vast span of time. The living and extinct organisms that Darwin observed in the flat plains of the Argentine Pampas, Pampas are vast grassy treeless plains in South America, and the Galapagos Islands sowed the seeds of Darwin's views on evolution. From old riverbeds in the Argentine Pampas, he dug up bony remains, fossils, of extinct mammals such as Toxodon, Macrauchenia, Pyrotherium and Thotherium, figure 5.3. The presence of Thotherium testified that a horse had been among the ancient inhabitants of the continent. It was the Spanish settlers who reintroduced the modern horse, Equus, to the continent of South America in the 16th century. Darwin wondered that a native horse should have lived and disappeared in South America. This was one of the first indications that species gradually became modified with time, and that not all species survived through the ages. Further, when Darwin collected the remains of giant armadillos and sloths on an Argentine pampa, he revealed the fact that, although they clearly belonged to extinct forms, they were constructed on the same basic plan as the small living armadillos and sloths of the same region. This observation initiated him to think of the fossil sequence of a given animal species through the ages and causes of extinction. Moreover, what Darwin had appreciated was that living species have ancestors. On traveling from the north to the south of South America, Darwin observed that one species was replaced by similar, but slightly different, species. In the southern part of Argentina, Darwin caught a rare species of ostrich that was smaller and differently colored from the more northern common American ostrich, Rhea americanus. This rare species of bird was later named Rhea darwini. It appeared to Darwin that species change not only in time but also with geoi, mf. Graphical distance, I in the f. Galapagos Islands Toxodon Thotherium. Darwin's scientific curiosity was greatly motivated by the many distinctive forms of life. The Galapagos consists of an isolated cluster of rugged volcanic ice Pyrotherium e. Lands in the eastern figure 5.3. Curious extinct hoofed mammals, ungulates, of South America. Pacific on the equator. Bones of these great mammals were found by Darwin on the flat about 600 miles west treeless plains of Argentina, after Volpe, 1985. Of Ecuador. One of the most unusual animals is the giant land-dwelling tortoise, which may weigh as much as 275 kilograms, grow to 183 centimeters in length, and attain an age of 200 to 250 years. The Spanish word for tortoise, Galapago, gives the islands their name. Darwin noticed that the tortoises were clearly different from island to island, although the islands were only a few miles apart. In isolation, Darwin reasoned, each population had evolved its own distinctive features. Yet, all the island tortoises showed basic resemblances not only to each other but also to relatively large tortoises on the adjacent mainland of South America. All this communicated to Darwin that island tortoises shared a common ancestor with the mainland forms. The same was true of a group of small black birds known today as Darwin's finches. Darwin observed that the finches were different on the various islands, yet they were obviously closely related to each other. Darwin concluded that finches were derived from an ancestral stock that had emigrated from the mainland to the volcanic islands and had undergone profound changes under the different conditions of the individual islands. Apparently, a single ancestral group can give rise to several different varieties of species. I.L.A. Galapagos tortoises feed on prickly pear cactuses. B. On islands with tortoises, a young cactus quickly grows a tall trunk, which lifts the succulent pads beyond the reach of the tortoises. 2. In the late 1830s, Darwin attended the meetings of animal breeders and intently read their publication. Animal breeders were conversant with the variability in their pet animals, and dwelled on the technique of artificial selection. Thus, the breeders selected and perpetuated those variant types that interested them or seemed useful to them. The breeders, however, had only vague notions as to the origin, or inheritance, of the variable traits. Darwin acknowledged the unlimited variability in organisms, but was never able to explain satisfactorily how a variant trait was inherited. Although Darwin criticized many aspects of Lamarck's theory, he did not deny that acquired characteristics can be transmitted. He realized that the nature of inheritance was unknown, but he devised a working hypothesis, called hypothesis of pangenesis equals origin from all, in 1868 to explain how acquired variations may be transmitted. G. Fortis e. Oqg fuliginosa. G. Conorostris. Dog diversity illustrates artificial selection. Pangenesis hypothesis. Darwin's pangenesis hypothesis assumed that all the organs, and perhaps all the cells, in the body emphifish s of an animal produced miniatures of themselves. These miniatures, called gemmules or pangenes, were shed into the G. difficilis, original colonizing species, bloodstream and carried to the sex glands. Camarhynchus nhun. V. C. Reproducti. Ve. Org. Scandens icrasirostris gans, the testes and ovaries where they newer assemble to C. popper. Form sex cells, the C. setacula eggs or sperms. Later, when a fertilized egg C. undergoes develop underscore. Meant, according to the concept, the pangenes present or responsible fourth particular features of the new Pallidus. C. Parvulus. On Cocos Island. Dash. I. Certhidia olivacea. C. Heliobates. Paneroloxias inornata. Individuals. In this figure 5.4. Darwin's finches. Manner, environment. Tall changes would produce modified organs, which in turn would produce modified pangenes that would transmit the change to the next generation. In year 1875, Galton made pangenesis hypothesis untenable by presenting several experimental proofs. He and others at later time made a series of experiments involving blood transfusions and later, transplants of ovaries between black and white varieties of rabbits and chickens. Gametes produced by the transplanted ovaries were consistent with the phenotype of the individual in which the ovary originated and not with the animal currently carrying the ovary. Blood transfusions had no effect on the gametes produced. These experiments readily demonstrated that the pangenesis hypothesis was incorrect. 3. Having explained the origin of variation, although incorrectly, Darwin wondered how artificial selection, a term familiar then only to animal breeders, could be carried in nature. There was no breeder in nature to pick and choose. In 1838, Darwin found the solution in Thomas Robert Malthus' book, An Essay on the Principle of Population, in which Malthus asserted that the reproductive capacity of mankind far exceeds the food supply available to nourish an expanding human population. Humans compete among themselves for the necessities of life. It, thus, occurred to Darwin that competition existed among all living things. Darwin then envisioned that the struggle for existence might be the means by which the well-adapted individuals survive and ill-adjusted are eliminated. Darwin was the first to realize that perpetual selection existed in nature in the form of natural selection. In natural selection, as contrasted to artificial selection, the animal breeder or horticulturist is replaced by the conditions of the environment that prevent the survival and reproduction of certain individuals. Thus, natural selection is a term serving to inform us that some individuals leave more offspring than others. It is not purposeful or guided by a specific aim. It does not seek to attain a specific end. Thomas Malthus, 1766-1834. Darwin-Wallace theory of natural selection. Darwin-Wallace explanation of the way in which evolution occurs may be generalized as follows. The change in species by the survival of an organismal type exhibiting a natural variation that gives it an adaptive advantage in an environment, thus, leading to a new environmental equilibrium, is evolution by natural selection. Thus, natural selection is a continuous process of trial and error on a gigantic scale, for all of living matter is involved. It includes the following elements. 1. The universal occurrence of variation. Variation is the characteristic of every group of animals and plants, and there are many ways in which organisms may differ. Darwin and Wallace did not understand the cause of variation, and assumed it was one of the innate properties of living things. We now know that inherited variations are caused by mutations. 2. An excessive natural rate of multiplication. Every species, in the absence of environmental checks, tends to increase in a geometrical manner. If a population of a given species doubles in one year and if there are no checks on its increase, it will quadruple the next year, and so on. Such a great reproductive potential of different species may be easily observed in nature. It has been estimated that a common Atlantic coast oyster may shed as many as 80 million eggs in one season. A salmon produces 28 million eggs in a season. A single pair of English sparrows would be the ancestors of over 275 billion individuals in 10 years if they and their descendants could reproduce at their natural rate without any check. Darwin calculated that even a pair of elephants, which are about the slowest breeding animals known, could, in the absence of any checks, have 29 million descendants at the end of 800 years. Thus, more organisms of each kind are born than can possibly obtain food and survive. Since the number of each species remains fairly constant under natural conditions, it must be assumed that most of the offsprings in each generation perish. If all the offsprings of any species remained alive and reproduced, they would soon crowd all other species from the earth. 3. Struggle for existence. Since more individuals are born than can survive, there is an intraspecific or environmental struggle for survival, a competition for food, mates and space. This contest may be an active kill or be killed struggle, or one less immediately apparent but no less real. Such as the struggle of plants or animals to survive drought or cold. Every species has an excessive rate of multiplication. 4. The consequent elimination of the unfit and the survival of only those that are satisfactorily adapted. Some of the variations exhibited by living things make it easier for them to survive, others are handicaps which bring about the elimination of their possessors. This idea of the survival of the fittest is the core of the theory of natural selection. 5. The inheritance of the mutations or recombinations that make for success in the struggle for existence. The surviving individuals will give rise to the next generation. And, in this way, the successful variations are transmitted to the succeeding generations. The less fit will tend to be eliminated before they have reproduced. Successive generations in this way tend to become better adapted to their environment. Negative, A positive. Natural selection. Figure 5.5. Diagram showing negative and meant, as the environment changes, further adaptations occur. The operation of natural selection over many generations may produce descendants which are quite different from positive natural selection. Their ancestors, different enough to be separate species. Furthermore, certain members of a population with one group of variations may become adapted to the environment in one way, while others, with a different set of variations, become adapted in a different way, or become adapted to a different environment. In this way, two or more species may arise from a single ancestral stock. Critical analysis of Darwinism. With the appearance of Darwin's book in 1859, a veritable storm of controversy broke out. Many people became interested in his presentation. The arguments for and against that developed were often heated. The concept of natural selection was opposed primarily on ethical and religious grounds. But the sheer weight of evidence and the logic of Darwin's presentation finally convinced a majority of the educated people. The concept of evolution affected many aspects of man's thinking, and it will continue to do so. However, the idea of the survival of the fittest of Darwinism has been erroneously interpreted by many to mean survival only by tooth and claw, an interpretation that has led in many cases to rationalizations for the attitude of every man for himself in social and economic affairs. In fact, the process of evolution is not all tooth and claw. Plants have evolved too, and there is no bloody competition between them. In all forms, plants and animals alike, a part of the selective process involves the action of inanimate nature, factors such as drought, storm, moisture, temperature, etc. Active competition between organisms is a part of the process, but as Walter Allais and others have pointed out, cooperation has, in many cases, been involved in survival. Natural selection results in the survival of those forms that are best integrated with the various factors of the environment in which they live. Certain other demerits of Darwinism. Besides its numerous merits, Darwinism has following demerits. 1. Darwinism or natural selection theory does not account for the beginning of organs, which may appear at first as the various rudiments having as yet no selection value. In other words, it remains concerned with the survival of the fittest, but not for the arrival of the poet Alfred Lord Fittest. Thus, to give rise to such specializations is elaborate mimicry. Tennyson wrote his poem Enter the Electri. See organ of the torpedo, etc. Y. Ch. of Apparent Memoriam in 1833. 25 years. Dvinth. D. Selecti. Before Darwin published The Advantage Only in the Perfected State, Natural Selection, Acting Only, Ogden of Species. It includes upon minute gradations toward perfection, seems inadequate. That the memorable line, nature same is true of so complex a specialization as the eye and its function, read in tooth and claw. In the vertebrates or in the insects and crustaceans. 2. Overspecialization in certain cases like extinct Irish deer in which huge antlers outweigh the entire skeleton, or the immense spiral tusks of the Jefferson mammoth, or the minute fidelity of certain mimicking insects such as Kallima, or huge dinosaurs of Mesozoic, all cannot be explained on the basis of continuous variations and natural selection. These organs or body structures should not have reached such a harmful stage, if natural selection was operating. However, such cases of overspecializations have been explained by Darwin on the basis of discontinuous variations or sports, which, according to him, do not play any role in evolution. 3. Natural selection cannot account for degeneracy. To say an organ is no longer useful and, hence, disappears, is to state the effect and not the cause. If under changed conditions a character built up by natural selection becomes a menace, the reversal of selection can accomplish its removal, but this will not suffice where the characteristic is an indifferent one. 4. One of classical objections to natural selection is that new variations would be lost by dilution, as the individuals possessing them bred with others without them. We now know that although the phenotypic expression of a gene may be altered when it exists in combination with certain other genes, yet the gene itself is not altered and is transmitted to succeeding generations. 5. Darwin indirectly accepted the Lamarckian idea of inheritance of acquired characters in the form of pangenesis hypothesis, which cannot be accepted in the light of knowledge of genetics made available in the present century. In year 1875, Galton made pangenesis hypothesis untenable by presenting several experimental proofs. He and others at later time made a series of experiments involving blood transfusions and later, transplants of ovaries between black and white varieties of rabbits and chickens. Gametes produced by the transplanted ovaries were consistent with the phenotype of the individual in which the ovary originated and not with the animal currently carrying the ovary. Blood transfusions had no effect on the gametes produced. These experiments readily demonstrated that the pangenesis hypothesis was incorrect. 6. Lastly, some persons have objected natural selection because it is essentially a materialistic doctrine, depending as it does purely on the laws of chance. Neo-Darwinism. Neo-Darwinism is a modified form of Darwinism. The Neo-Darwinians like T. H. Huxley and Herbert Spencer of England, D. S. Jordan and Asa Gray of United States, and E. Haeckel and A. Weismann of Germany believe that natural selection has accounted everything that is involved in evolution. In their dogmatic belief they went further to secure enough proof towards natural selection. Certain Neo-Darwinians, such as Weismann and his followers, rejected Darwin's theory except its principal element of natural selection. These Neo-Darwinians, though distinguished between germplasm and somatoplasm of living organisms in their germplasm theory, yet they could not appreciate the role of mutations in evolution. While Darwin believed that the adaptations result mainly by a single source, i.e., natural selection, Neo-Darwinians thought that adaptations result from multiple forces and natural selections is only one of these many forces. Neo-Darwinians also believe that characters are not inherited as such but there are character determiners, the determinants or biophores, which control only the development. The ultimate character would result out due to the interaction of the determiners, activity of the organism and the environment during development. Thus, Neo-Darwinism was incomplete and partly wrong because it lacked present understanding of genetics. Maturation of Neo-Darwinism into modern synthesis. During later half of 19th century and at the beginning of 20th century, many important ideas have been forwarded for explaining the inheritance of variations from one generation to successive generations and also for explaining evolution. Thus, the rediscovery of Mendel's laws of heredity by Correns, de Vries, and Chirmich in 1900 made it clear that 1. The factors given to the offspring by the parents do not mix, but are segregated, and 2. In more than one pair of contrasting characters are considered in the same cross, the factors responsible for these are inherited independently. Likewise, mutation theory of evolution of Hugo de Vries, 1886-1887, stated that new species arise by sudden changes or steps called mutations rather than by gradual processes. According to de Vries, it was mutations and not selection that should be considered as the primary factor in evolution. Further, the studies of Wagner, 1868, suggested the role of geographic or spatial isolation in the formation of every species, race, or tribe of animals or plants on the Earth. Even more, certain population geneticists realize that the actual physical struggle between animals for survival or the competition between plants for space, sun and water is much less important as an evolutionary force than Darwin believed. The evolution of any given kind of organism occurs over many generations during which individuals are born and die, but the population has a certain continuity. Thus, the unit in evolution is recognized not the individual but rather a population of individuals. A population of similar individuals living within a circumscribed area and interbreeding is termed as a deme or a genetic population. The next larger unit of population in nature is the species, composed of a series of intergrading demes. Further, in a population, the relative frequencies of the genes will remain constant from one generation to the next. 1. If the population is large. 2. If there is no selection for or against any specific gene or allele, i.e., if mating occurs at random. 3. If no mutations occur. And 4. If there is no migration of individuals into or out of the population. The operation of the Hardy-Weinberg principle will result in maintaining a given gene frequency in a population. Thus, a population undergoing evolution is one in which the gene pool is changing from generation to generation. The gene pool is the sum total of all the allelic genes in a population, and the gene pool of a given population may be changed, 1. By mutation, 2. By hybridization, that is, by introduction into the population of gene from some outside population, or 3. By natural selection. Recombination brought about by crossing over and by the assortment of chromosomes in meiosis may also lead to new combinations of genes and phenotypes with some specific advantage or disadvantage for survival that would be reflected in a change in the gene pool. All these modern understandings in cytology, genetics, cytogenetics, population genetics, and evolution gave way for the formulation of a coherent theory called modern synthesis around 1930s, by S. Wright, H. J. Muller, Th. Dobzhansky, R. B. Goldschmidt, J. S. Huxley, R. A. Fisher, J. B. S. Haldane, Ernst Mayr and G. L. Stebbins. 3. Modern synthetic theory. The modern synthetic